So let's talk about the different methods of sexual reproduction, or we can say different methods of plasmogamy. Okay. So when we talk about different methods of plasmogamy or sexual reproduction, in this first we talk about gametic fusion or planogametic copulation. In this, gametes are fused directly. So here it can be isogamy, anisogamy, or oogamy. It can be of any of these three types. Now another second type is gametangial contact. Now here it is known as gametangial contact because here only two gametangia are going to come in contact. Suppose this is the particular multinucleated hyphae of plus strain or male, and this is a multinucleated hyphae of minus strain. Now what happens? This both produce gametangia, so it produces gametangia like this, and this is going to produce gametangia like this. Now this gametangia is known as antheridium, and this is known as oogonium. Now they have particularly nucleus. This also have nucleus. Now a fertilization tube developed from the antheridium and enters the oogonium. So next, diagrammatically, we will observe such event. So here, particularly fertilization tube develops between male and female gametangia or from antheridium and towards the oogonium. So here, this is the way of plasmogamy. And after plasmogamy by fertilization tube, there will be occurrence of karyogamy. And if karyogamy occurs here, you will see that the zygote is going to form. Here, this zygote is diploid, and this zygote is also known as oospore. So be remember this oospore as the thick-walled zygote, and that is particularly diploid. After this, meiosis will occur, and haploid spores are produced. And these haploid spores germinate or undergo germination and form new mycelium. Okay, so this way gametangial contact occurs, and it is known that this gametangial contact is found to occur in case of oomycetes. So because of this, we can say these oospore are also produced in case of oomycetes, that is a lower fungi. Next to that, third type of plasmogamy, which we can study, that we are going to discuss. Then we are going to talk about a third type of plasmogamy, that is gametangial copulation. In this gametangial copulation, dear student, it is known that there is a two hyphae, and this hyphae developed some swelling structures. And these both hyphae are multinucleated, means they are of lower fungi. Okay, and both are known as progametangia. Now this progametangia convert into gametangia. So both will be modified like this. This is male gametangia, this is female gametangia. Now both undergo plasmogamy. And in plasmogamy, these both gametangia fuse directly. So due to the fusion of them directly, a multinucleated body is formed here. And after multinucleated body, then next you will find that karyogamy take place. And when karyogamy takes place, zygote is formed, and that zygote is known as zygospore. It is also called zygote. And after the zygote undergo meiosis and produce haploid spore. And this haploid spore undergo germination, and after germination it will produce mycelium. So dear student, here this is the way, and here the direct fusion of this two gametangia occur. Because of this we call it gametangial copulation, whereas in the previous one there is a formation of contact tube, that is a fertilization tube, and contact occurring, so it is known as what? A contact layer. But here direct fusion is occurring, so it is known as copulation. And this copulation is known to occur in case of zygomycetes. Okay. And after this gametangel contact, we are going to talk about next one, that is spermatization. In this fourth type of plasmogamy, that is spermatization. What will happen? See, suppose this is a female hyphae, this is a male hyphae. And this male hyphae produce non-motile spore or non-motile male gametes, sorry. Now this male gamete reach to the female receptive hyphae. This is female receptive hyphae. And with this female receptive hyphae, receive non-motile male gamete and fusion occur and zygote will be formed. This zygote undergo meiosis again here and form haploid spore. And this haploid spore on germination give rise to the new mycelium. Okay, so that is the thing about spermatization. And after spermatization, next type of plasmogamy that we are going to study, that is somatogamy. In somatogamy, dear student, that in this sex organ are not formed. Okay, sex organ are non-formed. And in this, what happens? Two mycelium come close to each other and get fused. So in this way, the ultimate fusion occurs, that is known as somatogamy. So these are the all types of sexual reproduction or all types of plasmogamy that occur during sexual reproduction have been discussed. Now we will talk about this again, we will take this term again during the classification. Okay. So dear students, after this particular different types of plasmogamy, we will see that there is a formation of fruiting body as well, in which the reductional division meiosis occur, as we have seen during the particular different type of sexual reproduction that lead to the formation of haploid spores. Now, after this, we are going to talk about classification, and in classification, it is known that the morphology of mycelium, then a mode of spore formation, fruiting body formation, form the basis of division of this kingdom into various groups, various classes. So there are these three important bases that you should remember. That is the structure of mycelium, morphology of mycelium. Second is mode of spore formation, and third one is the mode of fruiting body formation. And this divide particular fungus into different classes, and that classes are Phycomycetes, Ascomycetes, Basidiomycetes, and Deuteromycetes. Now, here in this, if we see, then we will study that these Phycomycetes are also divided into two groups. These Phycomycetes are also divided into two groups, that is Oomycetes and Zygomycetes. Okay, this Phycomycetes is known as algal fungi, when these Oomycetes, if we talk, they are also known as algal fungi. This is also known as conjugation fungi. Okay, so these are the different common names of this particular algae. Ascomycetes is also known as sac fungi. This is a common name. For Basidiomycetes, the common name is club fungi. Why these common names are given, we will discuss when we will study each class in detail. And Deuteromycetes are known as fungi imperfecti or dustbin fungi. So we will study this particular classes in detail. That time you will understand why they are named like this. So first we talk about some of the MCQs. The unique kingdom of heterotrophic fungi, direct question, heterotrophic organism, direct question you will find, and you know that they are fungi. Then the second question which we discussed, that is wheat rust disease of fungi is caused by, you know that wheat rust is particularly caused by Puccinia, but we will discuss this also in detail during the Basidiomycetes. Then cenocytic hyphae are found in the lower fungi, and cenocytic means multinucleated hyphae without septa. So the answer becomes option 1. Then the cell wall of fungi is made up of, dear students, the cell wall of fungi is made up of chitin, and that chitin is a polymer of N-acetyl glucosamine, as we have discussed. So till now we have discussed about this particular one, and now we are going to talk about the classes in detail. So let's start with the classes of fungi. So we are dealing with the first class initially, and the first class of the lower fungi, that is Phycomycetes. In this, we come to know that the members of Phycomycetes are found in aquatic habitat. Actually, these members of Phycomycetes, if we talk about habitat, they are aquatic in habitat. Apart from this, they are also found on decaying wood in moist and damp places. In moist and damp places also they are found. And they are considered as obligate parasite of plant. Okay. Then these Phycomycetes are also known as algal fungi, as we have discussed. When we see the mycelium of this particular Phycomycetes, we have studied that in lower fungi the septa or crosswall are absent, so mycelium or hyphae become aseptate. And if that aseptate, then the whole mycelium or hyphae will be multinucleated, and the term for the multinucleate condition used, that is xenocytic. Then you can say this is the particular hyphae, a fungus hyphae structure is there, forming a whole mycelium with the multinucleated characteristic. Apart from this, when we talk about in asexual reproduction of this Phycomycetes lower fungi, then it is known that it occur by the zoospore which are motile and aplanospore which are non-motile. You can see these are the zoospore which have a 4 flagella. One more thing is there, the spores, this zoospore or aplanospore, as we have discussed in asexual reproduction, they are produced endogenously within sporangium. We have discussed that both are formed in favorable condition. The difference is zoospore have a flagella, so they are motile, and aplanospore do not have a flagella, so they are non-motile. And one more difference is that zoospore are zoospore are wall-less, and aplanospore are thin-walled. This is the also a structure which is showing a release of sporangiospore. Dear student, as you can see here, this is a particularly a sporangiophore, and this is the sporangium, and from this sporangium the spores or sporangiospores are getting released. Okay, so this body is known as sporangium. Okay, then in the zygomycetes, in this phycomycetes, we will see the formation of zygospore by the fusion of two gametes. These gametes may be isogamous or anisogamous or oogamous in morphology. Then some common example included are mucor, rhizopus, and albugo. Albugo is a parasitic fungi on mycelium. And you can see here the isogamy and isogamy is shown here, and here in isogamy both the gametes are almost similar in size. As we have discussed, they are morphologically similar because size shape is similar, but in physiology they will be represented as male and female, so physiology is different and they are getting fused, so they are isogamy. In an isogamy both are dissimilar in size, so morphologically as well as physiologically they are dissimilar and they are fusing. And in heterogamy there is also oogamy, male gamete is motile and female receptive hyphae is there and the fusion is occurring. So mucor is found to be grown on this particular braid as well. Rhizopus is seen like this as a particular black bread mold. Okay, and you can see this as albugo. So this is generalized information about this particularly phycomycetes according to NCERT. But dear students, if we study, these zygomycetes are divided into two groups, that is oo and these phycomycetes are divided into oo and zygomycetes. So if first we are going to talk about oomycetes. These oomycetes are also known as algae fungi. Now, if this is a type of phycomycetes, and obvious, this is also aseptate, and this is also particularly multinucleated. Now, apart from this, when we talk about this oomycetes, in this particularly, we see that one of the important point is that in this the cell wall of this oomycetes is made up of specifically the cellulose or glucose derivative. Otherwise, in majority of fungi, the particular or remaining all classes, you will see that the particular cell wall is made up of a chitin. Apart from this, we will come to know that there is a different type of reproduction, and in reproduction also there is a asexual reproduction and sexual reproduction. When we talk about asexual reproduction, in case of oomycetes, this asexual reproduction occur by formation of juice spore, by formation of juice spore. Whereas when we talk about sexual reproduction, this sexual reproduction is occur by gametangial contact. Now, this is the some of the information with respect to oomycetes. Mainly we talk about the examples of oomycetes fungus, that is lower fungi. And in that first example which we are going to study, that is Phytophthora infestans. And when we talk about this Phytophthora infestans, this Phytophthora infestans cause late blight of potato. Late blight of potato. Late blight of potato means there is a different colored patches or blights on the potato in the late in the season, and due to this late blight of potato, one havoc produced in the Ireland that is known as famine of Ireland. And during that time, that is in 1845, what happened, the availability of this potato was reduced due to this infection. Apart from this, second example is of Albuco candida. And this Albuco candida or Cystopus candidus, and this Cystopus candidus cause white rust of crucifers. White rust of crucifers, in which white spots are developed on the particular the members of Cruciferae family, specifically on the leaves. Apart from this too, the third example is Pythium debrinum. So when we talk about this Pythium species, that is Pythium debrinum, this Pythium debrinum specifically cause damping of disease. And in this damping of disease, what happens? The stem get broken up. Okay, and this damping of disease is found in case of tobacco and some vegetable crops. Okay, so these three examples are important. Apart from this, one fourth one, that is saprolegnia. There is no need to study detail, just you should know that saprolegnia belong to this oomycetes. Now, after oomycetes, next we are going to talk about the second type of phycomycetes, that is zygomycetes. And when we talk about the zygomycetes, in zygomycetes, Specifically, these zygomycetes are also aseptate because they are lower fungi and multinucleated means xenocytic mycelium is found. When we talk about particular reproduction, their cell wall, first the cell wall is made up of chitin. When we talk about reproduction, reproduction is of two types, that is asexual reproduction. And when we talk about asexual reproduction occurs by formation of sporangiospores, that is specifically aplanospore. Then when we talk about sexual reproduction, it is known that in case of sexual reproduction, it occurs by gametangial copulation. Now, in this, there are some examples, and in examples of zygomycetes, if we take, first comes that is the rhizobus and mucor. Now, when we talk about this, they are known as bread mold. They are known as bread mold, and they prefer to grow on bread. They prefer to grow on bread. Okay. And in this, Rhizopus is known as a common bread mold. It is found commonly growing on bread, so it is known as common bread mold. And when we see the tip of mycelium of this Rhizopus, it is black colored due to what? The black colored spore. So this fungus like a black color, so it is also known as black bread mold. One species of mucus is also there, and this mucor are also known to be dung mold. And in that one species is there, that is mucor mucedo, that is known as pin mold. We remember it is not pink, it is pin mold. And apart from this, the another example which can be observed, that is pilobolus. Okay, so that pilobolus is also an example of this zygomycetes. So this two are found, and we have studied that these zygomycetes are also known as conjugation fungi. So after studying this phycomycetes lower fungi in detail, next we are going to talk about the another group of higher fungi, that is ascomycetes. Ascomycetes are commonly known as sec fungi, and the ascomycetes are unicellular. So yeast belongs to which class? Ascomycetes. And the genus of yeast is Saccharomyces, a known genus, common genus. It is not that they are only unicellular, they can be multicellular as well as that's the penicillium. And we have studied about that in the spore. This is the yeast. This is the penicillium. We have studied this particular branching and diagram of this penicillium. Then this ascomycetes are considered as a saprophytic decomposer, parasitic or coprophilous. Coprophilous means what? It is growing on the dung. Then this is a coprophilous fungus which is growing on the particularly dung. Okay, so this particular nutrition, this different type of nutrition can be found in case of ascomycetes. Apart from that, when we see their mycelium, this is a higher fungi, so their mycelium is branched and septate. Their mycelium is branched and septate. And branched and septate mycelium, then if we talk about the reproduction, and in reproduction we come to know that there is a asexual spore, mitospore, and conidia are produced here. We have studied that conidia are produced exogenously on the special hyphae, that is conidiophore, specifically on the sterigmata in a basipedal order that we have discussed in that particular type of spore. Here you can see this is the septum, this is the hyphae, this is the mycelium, and septate mycelium is found, and branched mycelium is found in case of Ascobicites. That this is the particularly arrangement of penicillium, and here you can see the asci are given. What are asci? We will discuss this in detail in the next slides. Conidia on germination produce mycelium, means conidia are the asexual spores, so obviously they are haploid produced by mitosis, so they will germinate and they will form a new mycelium of this Ascomycetes. Okay, now in this Ascomycetes we will discuss that how they reproduce. Okay, and before this thing we will study how particularly they carry out the different things. So when we see the life cycle, in life cycle we will come to know that, or specifically when we talk about reproduction, asexual reproduction we have discussed, that is by conidia, but we have to again discuss the sexual reproduction in case of Ascomycetes. Now when we go on study the sexual reproduction, in sexual reproduction, we come to know that it is by somatogamy and gametangial contact. It is by number one, somatogamy, and number two, that is gametangial contact. Okay. Now here in this first we are going to study this somatogamy. As we have studied during plasmogamy, here without sex organ formation, the sexual reproduction occur. So when we see the diagrammatic view of this particular somatogamy, we will see that how this occur. So this is the particularly higher fungi. One is plus, one is minus. Plus show the male one, minus show the female one. Now this both come close to each other, and after coming close to each other, they undergo plasmogamy. When we see the plasmogamy, the dikaryophase is observed. And this dikaryophase with a particular cell showing a dicaryonic condition. This dikaryophase with the two haploid nucleus is short-lived. And after this, when we talk about our next diagram, we'll come to know that after plasmogamy, dikaryophase will observed, and the cells with the two nucleus will give rise to the one extension, and that extension will be like this, and this particular hyphae is known as ascogenous hyphae, and this structure is known as ascus. Now in this ascus, two nucleus which was here in dikaryon that goes here, and after reaching ascus the karyogamy occur. When karyogamy occur within ascus, the diploid nucleus will be formed. And when this diploid nucleus is formed, this diploid nucleus undergo meiosis. And when meiosis occur, then four ascospores are produced. So four ascospore are produced within the ascus. So we can say they are endogenously produced sexual spore. Now one more thing happens, the surrounding hyphae which was present that keep on growing, and when the surrounding hyphae keep on growing, they form covering over the particular body, and in this particular ascus this is formed. So this whole body is known as a fruiting body of ascomycetes, that is ascocarp. After this, when the ascocarp will be broken up, the ascospore will be released, and ascospore, after release, they will give rise to the what? A new mycelium. So this is the way of somatogamy. So after this somatogamy, the ascospores are released. So because of the formation of ascus-like structure, they are known as what? Particularly ascomycetes. And here, the important one is ascospores are produced. And when we talk about this ascospore are sexual spore, but they are produced endogenously. Whereas if we talk about asexual spore in ascomycetes, it is conidia and it is exogenously produced. So when we talk about ascomycetes, they have a exogenously produced asexual spores and endogenously produced sexual spore. Sometime in ascus, the mitosis occur in case of 4 ascospores and the 8 ascospores are produced. Now we will be dealing with some of the examples of this ascomycetes one by one. But before that, you should understand that during the sexual reproduction in a dikaryotic phase, with ascogenous hyphae is formed. Sexual spore are ascospore which are produced endogenously in a sac-like structure, and C compatible yeast cell fusion will occur. The two nucleus will come, fuse, zygote will be formed. After this, meiosis and mitosis will occur, and ascus contain 8 ascospores. So karyogamy and meiosis occur in SI or ascus. This SI are arranged in different type of fruiting bodies. So I want me to make you understand this. This SI or ascus are arranged in different type of fruiting bodies called a ascocarp. You can see this is the fruiting body. Here you can see this is showing a fruiting body, and in fruiting body this the enlarged view of this is shown in like this, and these are the ascus. In this so many ascus you will find a presence of 8 ascospores which are arranged linearly like this. Okay, so these are the ascospores within ascus. Okay, So the globose ascocarp without opening is called clistothecium. See, like this, a closed, completely closed ascocarp is known as clistothecium. And here the clistothecium ascocarp diagram is given. You can see here as well that ascospores are produced within the particular globose structure and covered by the clistothecium wall. So it is giving a close appearance. So it is known as clistothecium. Then sometimes in some of the particularly fungus, the ascocarp is flask shaped, and this flask shaped ascocarp has an apical opening. So this type of ascocarp is known as perithecium, whereas cup or saucer shaped ascocarp is called as apothecium. So you can see here this is the perithecium, and in perithecium you can see this is the opening, and within that you have a asco particular ascus, and within ascus we see the presence of ascospores. Apart from this, the apothecium is a flask or saucer shaped, and you can see this is found saucer shaped particular apothecium. Then if we talk about the examples of ascomycetes, some examples of ascomycetes are aspergillus, claviceps, penicillium, and neurospora. Now, some importance with respect to all this we have to discuss, then we will go further. Here, the diagrammatic view of aspergillus is like this. The claviceps somewhat look like this. Then penicillium look like this. Then neurospora look like this, and it is pink in color. We will study that it is known as pink mold. So neurospora is used in extensive biochemical and genetical work, and due to this, neurospora is also known as Drosophila of plant kingdom. Now, we will particularly discuss some of the examples of this particular fungi in detail. And in this, first we are going to talk about examples of ascomycete. And in this, first we are going to talk about penicillium. Penicillium. Penicillium is known as blue or green mold. And when we talk about this, we come to know that this penicillium is a ascomycetes fungus. From the one species of penicillium, that is Penicillium notatum, penicillin drug was obtained. Obtained by Alexander Fleming. Now, what Alexander Fleming did? Alexander Fleming was working on one bacterial culture, and one day he forgot to wash this petri dish, and in the particular petri dish, he was when he left the particular house, he seen that the particularly whole petri dish was filled with the bacteria. But when he came after the that particular work, he said there's one dark colored body is growing here, and it is inhibit the growth in this area. This is known as inhibition zone. Means it is a petri dish of whole bacteria. Petri dish is here, okay, completely filled with the bacteria growing. But after he come back, he see that in this area the bacteria are not growing due to the presence of this structure. And later on he found that this is nothing but fungus, which is a releasing substance which inhibit the growth of bacteria in this particular region. And later on, he found that this fungus is nothing but it is a Penicillium notatum. And from this Penicillium notatum, the chemical which inhibit the growth of this particular bacteria was identified and that was named as penicillin. Okay. Now, this penicillin drug was discovered like this, and for this, Alexander Fleming was awarded Nobel Prize. But nowadays, there is a more need of penicillin. So, nowadays, penicillin is obtained from Penicillium chrysogenum. So now commercially it is from Penicillium chrysogenum. Okay. Now this discovery according to the Alexander Fleming is serendipity. This discovery of penicillin is by chance discovery. So it is known as serendipity. Okay. So this is about what penicillin. Now after this, next we are going to talk about some of the another particular, yes, one even more important point with respect to penicillin. You know that you should note that it is also used in making cheese. Like Penicillium camembert is that is used in formation of camembert cheese, and Penicillium roquefort species is there that is involved in formation of roquefort cheese. So this Penicillium species are also used in the formation of cheese. Then next we talk about a second example of this particular Ascomycetes. And the second example of this is Aspergillus, one of the known example. When we talk about this Aspergillus, it is black colored or brown colored mold. And it is also known as blacky smoky mold. Now, when we talk about this particular Aspergillus, we have to talk about some of the species of Aspergillus. And when we talk about some species, one species, a known species that you should remember, that is Aspergillus fumigatus. It is known that when we talk about Aspergillus fumigatus, it is a species that causes many diseases in human and cattle, and it is known for causes a disease aspergillosis. Apart from that, one more species is known, that is Aspergillus flavus, and this Aspergillus flavus prefer to grow on stored food. This prefer to grow on stored food like groundnut, cashew nut, etc. And it is also preferred to grow on fodder. It is known that it secretes toxic substance, that is aflatoxin. And aflatoxin is the substance which is carcinogenic, that is cancer causing, and they cause a liver cancer. Apart from this, third species that you should remember, that is Aspergillus niger. Majority time question come from this species. And this Aspergillus niger is known as weed of laboratory. Now, why it is known as weed of laboratory? Because it contaminate, is known as weed of laboratory because it contaminate laboratory cultures as well as foodstuffs. So because of this, it is known as weed of laboratory. So this is the another example. After this, we are going to talk about next example, that is Claviceps, which is Ascomycetes fungus. Claviceps. Just correct it. This Claviceps fungus is also example of this Ascomycetes. And in that one species is that is Claviceps purpura. That Claviceps purpura cause record disease of Bajra and rye. And in this particular fungus release one substance that is known as ergotin. And it is known that if ergotin is eaten by a pregnant lady by mistake, that pregnant lady may undergo miscarriage or expulsions of fetus can occur. Then next we talk about fourth example, that is Neurospora crassa. When we talk about this Neurospora crassa, it is known as red or pink mold. It is red or pink mold. And it is also known as Drosophila of plant kingdom. Why? Because it is known to use used for study of genetics and biochemistry in plant kingdom. So this is the one of the example. One more example that you should remember, that is Erisiphe. And that Erisiphe cause one disease, that is powdery mildew on plants, in which powdery coating occurs all over the plant body. So these are the some of the particular Ascomycetes fungus examples. Next to that, yeast are also placed in this particular one. Yeast are also example of this Ascomycetes. And in yeast, it is known that yeast are of different type, as you have studied. These are first of all unicellular. And in this, there are different example in which One of the known species is the Saccharomyces cerevisiae. Saccharomyces cerevisiae. And this Saccharomyces cerevisiae is known as baker's or brewer's yeast. And this Saccharomyces from this riboflavin, that is vitamin B2, is also obtained. Okay, so this is about the particularly yeast. Apart from this, Gibberella fusicorloi is also example of this Ascomycetes, and from this gibberellin, that is plant hormone, is obtained. Okay, so this is the one of the important stage. Okay, now after this we will again discuss about the Gibberella fusicorloi by relation with the Deuteromycetes when we will study Deuteromycetes. Okay, so I hope you have understood this particular part. Now we will be talking about the further part here, and then here there are many members of morels and truffles which are edible and are considered delicacies. So if you get a question of edible Ascomycetes, then we remember that these morels and truffles are the edible Ascomycetes. Then we talk about the next group, that is Basidiomycetes, which are also known as club fungi.